All right. Mic check, check, check. Right. All right. So we're moving on to chapter 21, congenital and genetic disorders. So going to be a lot of kind of some DNA talk in here, and we're going to kind of try to define some terms and hopefully make this a little bit easier for you. So what is the genetic information? So you kind of remember from A&P, our nucleus of our cell contains our DNA. That's kind of like the blueprint for who you are, and it's stored in our nucleus. So for humans, we've got 23 pairs. So that's 46 chromosomes, 23 paired together. 22 are known as basically autosomes, or autosomes are just the genetic code that makes you you, and one pair is always going to be the sex chromosome. So female is XX, male is XY. And in that, a karyotype is basically a visual representation. This kind of whenever you see people picture DNA or picture chromosomes, you're kind of picturing them looking basically a little bit like this shape here, like this helix shape. This is kind of a bad drawing, but that's what a chromosome kind of looks like. It's this karyotype is this visual representation of this here. So moving on, genetic control. So some definitions to define kind of the first part of this chapter: genotype and phenotype. So what genotype is basically all the cells of your body, of an individual's body, contain the same chromosomes or genes for the same traits. So genotype would be just like that blueprint that you have as an individual of your genes that make up kind of the same thing. And phenotype, what's talked about in a minute here, is the expression or the effect of that gene on you, like the visual representation or the effect, basically. Skin color, hair color, how the body presents it. And then one other definition, kind of the highlight from this, is the word allele. Allele gets a little bit confusing too. I mean, a lot of this stuff gets a little bit confusing, but what allele is basically is a representation, or your genes all have multiple functions, and the functions of that gene are known as allele. And then how they present would be the phenotype, and the genotype would just be the organization in each of your cells. So you imagine allele is just kind of the name for the gene and what it does. So if you have a gene that is skin color, that's an allele for skin color. A gene for blue or green eyes, that's the allele that we get from our parents. The genotype is the organization into chromosomes, and phenotype would be what expression does that give. So hopefully that makes sense. So talking about congenital disorders, they can vary. Usually it could be a mutation from mitosis during utero, while after conception, before birth. Sometimes they're also called congenital anomalies. Anomaly means something out of the norm or abnormal, and disorder would kind of mean the same thing. So most time congenital disorders are present at birth. Doesn't always mean that someone can have a congenital disorder that doesn't show at birth, doesn't have like basically a phenotype, a visual representation of that, but they can have the genetic or congenital disorder. And then inherited disorders can be caused by that sort of stuff. And that's where that phenotype kind of comes into the congenital disorders, the expression of that gene. So the genotype is the blueprint, and there it is. The alleles is more talking about what we get from our parents and the function that those genes have. And then phenotype would be that physical expression of a gene. And again, at birth, you maybe not have any. So examples of congenital disorders, there's talked about, this is on page 572. There's different types. Like I get a single gene from one of my parents, and it carries this thing. So Huntington, Carrera, polycystic kidney disease. And then there's things that are recessive and dominant. We're going to talk a little bit about what's recessive genes and dominant genes. And then there's some X-linked, dominant, X-linked, recessive. Those are some of the big ones. And then there's multifactorial, means there might be multiple genes involved, multiple things involved. And then chromosomal disorders are usually an extra chromosome or a missing chromosome, something like that. So just kind of as a reminder, this question is just asking, hey, as a basics, what is a total number of chromosome pairs in humans? And pause for a second. Okay. The answer is 23. 46 chromosomes, 23 pairs. They pair together. What is the visual representation of chromosomes arranged in order of size called? This is a visual representation. So these definitions are tough, I'm telling you, but pause. So this is karyotype. Karyotype is their visual representation, that little hexagonal kind of X shape for chromosomes that you see. That's how they're arranged. The genotype is kind of what they look like and what their function is, like identifying it. And then phenotype is that visual representation of the gene or the chromosome. What does it do visually to a person? Eye color, hair color, skin color, like visual, like what is it contributing to? So there's congenital disorders, again, sometimes called congenital anomalies or chromosomal anomalies. This is usually some kind of mutation or error in the DNA replication during meiosis and sometimes mitosis after conception. But meiosis is the splitting of sex cells. So they don't make exact copies of themselves. They make like a half genetic information. So, right. So if you remember a little bit, mitosis is the splitting that our cells do all the time, right? So just as a kind of refresher, mitosis is you got your cell, it makes exact copy of its DNA, the 46 chromosomes, and makes two daughter cells that carry the 46 chromosomes, the 23 pairs. But for meiosis, we have a sex cell here. So we, sperm or egg, whatever for female. So sex cell, when it makes copies of itself, these are non exact genetic copies. They only have 23 pairs, or they only have 23 chromosomes, excuse me, not pairs. So they only have one half the genetic information or the genotype, basically. They have half of it. So whatever, that's because what's a sex cell going to do? Its goal is to merge with the opposite sex cell and make a baby. That's a whole new genetic information. 23 chromosomes from mom, 23 chromosomes from dad. That's meiosis. So just kind of refreshing on the A&P there. So again, there's different types of disorders. Teratogenic disorders, it's kind of talked about on page 573, but this could be something that causes damage during the fetal development phase. So, of course, we've always identified different things that are harmful for people to do during pregnancy. They talk about a lot of stuff that women should do, drink alcohol, smoke cigarettes, teratogenic agents that are going to cause issues. And then there's also chemicals that people would recommend women of childbearing age to avoid. Multifactorial disorders, there's a large number that are in this type, and they often have a family history. Um, and there can be an environmental component, but these are more complicated. So you can kind of leave these. One of the things the book highlights is a Marfan syndrome. It's a skeletal deformity. The book highlights that on page 574. That would be a multifactorial disorder. So I think probably your take home from this slide is really the teratogenic agents are going to be things to avoid during pregnancy that have been known to, cause fetal development issues, that sort of thing. And overall, congenital disorders, it's good to keep in mind that they can not just result from hereditary, just inheriting it from mom and dad, but they can result from things like premature birth. This means that there's lacking fetal development in the embryonic phase. And so cells may mutate, things may not grow effectively. Difficulty during labor and delivery, especially in the vaginal or even C-section, really, too. Some things can be difficult and cause hypoxia in the child, and that can lead to cell damage and that sort of stuff. And then cerebral palsy is kind of an example of that. So on to kind of just the genetic disorders that are more cut and dry. You kind of got big three categories: single gene disorders, chromosomal disorders, and multifactorial disorders. So what type of just as a refresher before we get into it, so what type of disorders are caused by an error during meiosis? So an error during the splitting making of sex cells, basically. This one's pretty easy, straightforward, but yeah, congenital disorders, excuse me. So think of it that way: a issue during meiosis, the production of sex cells in a person can carry a congenital disorder. So single gene disorders, they get broken down in a couple of categories. Basically, they're classified by how they're inherited. So recessive, dominant, X-linked. So it just depends. We'll talk about the difference between recessive and dominant genes, but there's some single genes that control a very specific function. And so one thing that gets inherited, especially in men, is color blindness. It's a very specific function. But there are other disorders that are going to have whole body systemic effects: cystic fibrosis, PKU. So there's a lot of stuff that can have multiple body effects, not just, one function to one part of the body. So kind of the first section talks about autosomal recessive disorders. So basically what it's talking about here is both parents must pass on the allele for the disorder. So again, allele is basically just matching a gene to its function. And mainly when they use the word allele, we're talking more about the parents' genes and trying to map it to children's, or to their children or their expected children or future children, whatever. So again, those are some hard definitions for sure: the phenotype, genotype, karyotype. So karyotype, again, is just a visual representation of the chromosomes. They look in that X kind of shape. Genotype is just saying, here's the list of genes and they're mapped in this way for this person. And phenotype is saying what's that visual representation. The word allele is talking about an individual gene and matching it to his function more based on the parent's genes. So what recessive genes are and what dominant genes are, to summarize really, is that recessive genes are genes when they're passed down, they tend to uh, be more hidden in the background. If they're paired with something that's dominant or big, they'll take a back seat, and then therefore they won't show. Basically, they're not going to show up in kids. And so we'll talk a little bit about that. But like say, um, I don't know them all off the top of my head, but let's just say that the gene for brown eyes is recessive, and mom has brown eyes. Oh, excuse me. And dad has blue eyes, and the blue eye gene is dominant. So between that mom and dad, you can use what we'll look at in a minute, a Punnett square, to kind of figure out what is the percentage your children are going to have what eye color. Um, but it's more likely that they're going to have a higher percentage chance of having the allele for blue eyes because it's a dominant gene versus the brown that's recessive. That's just an example of what they're talking about here. So for autosomal recessive disorders, basically because they're recessive, it's for the child to have this disorder, both parents would have to have that recessive allele for a particular disorder. So parents may be completely unaffected by this disorder. They may be a carrier for this disorder, which the word they kind of use to describe that is heterozygous. So basically, they would have one normal gene and one disease gene, and it's a recessive gene, so it doesn't cause or manifest the disorder. It doesn't cause a phenotype, doesn't manifest the disorder in that person, but they're now a carrier for it. And that means that they can have the potential to pass that gene on, that recessive gene on to their children. And so in recessive disorders, usually it's going to come from parents that are homozygous, which means that uh, or a homozygous child is basically just saying that they have the defective gene and it's manifesting. So for autosomal recessive disorders, things to kind of keep in mind is it affects male and females equally. And so most likely if someone's homozygous, that means they got both the recessive genes. That child would have the disorder. If they were heterozygous, that would mean they wouldn't have any signs of disease and they're likely a carrier. So that's where like we talk about the Punnett square comes into play. And so I've got a video that hopefully kind of summarizes this here in a second. But what they're basically saying is that, and I'll try to kind of articulate this a bit for you, but what they're basically saying with these recessive disorders or any disorders in general is that there's a carrier parent, right? They're showing you here. Basically, the parent has a big C, which means it's the dominant gene. And in this case, a recessive disorder means that the disorder follows a lowercase g. I didn't make that a very good lowercase g. Stand by. So we make that a little bit smaller. So this is the recessive disorder. Because it's a little c. It's a gene. Okay. And so if you look at this, you're imagining these are two carrier parents that have a big C, little c, basically. So they both are carriers for the disease. And so what a Punnett square does is basically you're bringing this information down. And the video I have will kind of help probably uh, show this. But basically, what they're saying here is that if I take this, I'm taking this big C down, little c down, and you kind of just follow this stuff over. So big C, right? Again, big C is going to come down, little c comes in. what's coming down from here. Coming down from this parent is little c, coming over from this parent, little c. So the little c represents the disorder, a recessive gene that causes whatever disorder. In this case, they're using an example of cystic fibrosis. So what that's basically saying, if I kind of backtrack here and erase some of this stuff, what they're saying here is that using a Punnett square, you can kind of determine the probability. So what the probability is, if both parents are carriers, is that they'll have a 25% chance for their kids to be normal, no carrier for it, no, because you can see that because they've got a 1 in 4 chance of having a kid that has the 2 big C's, the dominant gene, no recessive gene, which means it doesn't carry the disease for cystic fibrosis in this case. And then they're going to have 50% chance to be a carrier because they're going to have a possibility of 2 out of 4. If they had 4 kids, just look at it that way, 2 out of the 4 kids would have the little c recessive gene that carries this disorder. And so they would be carriers. And then the way the Punnett square works, we saw that then there's going to be 1 child, 1 in 4 child is going to have 2 little c's, which are the recessive disorder, cystic fibrosis. So they're going to be affected by this. Again, this isn't one-to-one. A Punnett square is used to kind of predict possibilities. It's always possible that there is a 25% chance they'll have, but at least this family could have 4 children and none of them have the gene to carry the disease. It's not a perfect scenario, but this just tells you probability basically. And then this is the same example they're showing us here. They're showing us there's a parent that has the disease. So they have the recessive gene, 2 little c's. They have cystic fibrosis. And then you have a parent that has 2 big C's, doesn't have the recessive gene. So you just kind of, again, go across. So this parent would share a little c in every box, and this parent would share a big C in every box. And basically this makes like 100% chance that all their children are going to be heterozygous, that they're going to be carriers for cystic fibrosis. Not that they'll be affected, but again, if they were to go and get married and have a kid with someone that was also a carrier, then they would have the 25% chance to have an effective offspring. So that's what a Punnett square is doing. So hopefully this video will explain it even better than me, because, I'm not perfect for sure. But
by the end of this video, you're gonna have a solid understanding of genotype and phenotype, and you'll be a pro at using the Punnett square for simple crosses. But first, let's define a few terms. When we're talking about genotype, we're talking about the genetic makeup of the organism, the genes. These genes can come in different forms, and we call these forms alleles. Let's take an example of height in pea plants. There are different alleles for height. You can have an allele for tall and you can have an allele for short. Then we have phenotype. A phenotype is the physical expression of those genes. So in our example of height, the phenotype is the actual physical characteristic of tall or short. It's that simple. So the genotype refers to the genetic makeup, and the phenotype is how those genes are expressed physically. But how do we get from genotype to phenotype? And can we make predictions of what offspring will look like? Well, to a certain extent, we can, using a simple tool called a Punnett square. A Punnett square is a tool that geneticists use to predict the possible genetic outcomes of a cross between 2 parents. It was first described in 1905 by the British geneticist Reginald Punnett. This dude named the tool after himself in his book Mendelism. I always wanted to describe something significant and call it something like the Samuel thing. Okay, I didn't get that far yet, but let's continue. Anyways, I want to show you how to use the the Punnett square. We're gonna do this by going back to Gregor Mendel's experiment. We're gonna replicate his pea plant experiments conceptually without having to wait for actual pea plants to grow. First, let's look at the possible genotypes and phenotypes related to the height of the pea plant. The capital T allele codes for tallness, and that's a dominant trait with pea plants. And the lowercase t allele codes for shortness, and shortness is a recessive trait. Since each pea plant will have 2 alleles in the gene for height, one from each parent, the possible genotypes are capital T, capital T, capital T, lowercase t, and lowercase t, lowercase t. Now, I grew up saying big T and little t, so that's what we're going to use moving forward. Makes it easier to say. But we also have fancier terms to describe this even further. If an organism has two of the same alleles, we call it homozygous. And if it has different alleles, we call it heterozygous. So our options are homozygous dominant with two big T's, heterozygous with a big T, little t, and lastly we have homozygous recessive, little t. Those are the three possible genotypes. What about the phenotypes then? Well, a homozygous dominant pea plant will be tall. A heterozygous pea plant will also be tall since the tall allele is the dominant allele. And a homozygous recessive plant will be short. And that's the only genotype of pea plant that will actually be short, because once the dominant allele is present, it's going to mask the presence of the recessive short allele. Now, for the experiment, let's say we have a tall pea plant and a short pea plant. And for this first experiment, the tall plant has a homozygous dominant genotype. So big T, and the short plant is homozygous recessive, little t. We're going to cross these two plants, and this is where the Punnett square comes in handy. We're going to put one parent's allele on the top of our Punnett square and the other parent's allele on the side here. So at the top, we'll put big T, and on the side, we're going to have little t for the short plant. Then what we're going to do is we'll fill in each box of the Punnett square with the alleles from the corresponding rows and columns. So we'll pull the big T's down and the little t's over. This is then going to give us all of the possible genotypes that the offspring of these two plants could have. So as we can see, the resulting offspring will all be heterozygous, big T, little t. And what will the phenotype of this F1 generation be? Well, they'll all be tall. And yes, that's exactly what Mendel observed. So far, so good. Well, let's do exactly what Mendel did. He continued on and crossed the F1 generation. So let's make a new Punnett square and put the parents on the top and the left. Since all of the F1 generation plants were heterozygous, we'll put big T, little t in both spots. And once again, let's fill in all the options. Bring down the allele options from the parent at the top and bring over the allele options from the parent on the side. And when we look at the resulting options, we see that 1 out of the 4 offspring will be homozygous dominant, big T. 2 out of the 4 will be heterozygous, big T, little t. And 1 out of the 4 will be homozygous recessive, little t. Another way of saying this is that 25% of the offspring will be homozygous dominant, 50% will be heterozygous, and 25% will be homozygous recessive. Now, what does this mean for phenotype? Well, again, the allele for tall is dominant. So once that's present, the plant will be tall. So basically, in this situation, the offspring has a 3 out of 4 chance, or a 75% chance of being tall, and 1 out of every 4, or 25%, will be short. That's what we'd expect, and guess what? That's exactly what Mendel observed and described. Man, I don't know about you, but I'm just fascinated by the fact that we can use a simple tool like the Punnett square to make pretty accurate predictions about the genotypes and phenotypes of offspring. Now, in this video, we looked at very simple crosses that were looking at just one trait, like height. We call these monohybrid crosses. In the next video, we're gonna look at a more complex.
Okay, so hopefully that video kind of helped to summarize it a bit more. And so I have another video that talks about explains another way of explaining it. I think this one's also helpful, so I take some time to watch this one as well.
Hi, this is Tom from ZeroToFinals.com. Today I'm going to talk about a concept called Mendelian inheritance, and specifically the inheritance of autosomal recessive and autosomal dominant conditions. So some genetic conditions exhibit a simple inheritance pattern called Mendelian inheritance. This inheritance only occurs where the disease is caused by a single abnormal gene on one of the non-sex chromosomes. So this is the chromosomes that are not the X or Y chromosome. So these chromosomes are called the autosomes. Two examples of diseases that are caused by a single gene passed on from somebody's parents are Huntington's chorea and cystic fibrosis. So the difference between these two conditions is that the gene for Huntington's chorea is dominant over a normal gene, and the gene for cystic fibrosis is recessive over a normal gene. So this basically means that in order to have the disease of Huntington's chorea, you only need a single copy of the disease gene because it'll override the other copy of the gene that you have that's normal. Whereas in order to have the disease of cystic fibrosis, both of your copies of the gene need to be the disease gene. Because if you only have one disease copy, the other copy that you have, which is normal, will override the disease copy. In this scenario, you're called a carrier because you don't actually have the disease, but you do have the capacity to pass it on to your offspring. So we refer to diseases like cystic fibrosis as autosomal recessive, and diseases like Huntington's as autosomal dominant. So now I'm going to go through how you actually calculate the risk of one of the offspring having an autosomal recessive condition. So in order to calculate the risk of passing on an autosomal recessive condition, it's really helpful to draw out an inheritance pathway. By using this method, you can always work out the risk of various outcomes based on the genetic status of the parents. So I'm going to use some examples in order to demonstrate this method that could help you in your exams and also in explaining things to parents in real life practice. Firstly, let's consider both parents are carriers of an autosomal recessive condition. Each parent has one disease gene and one normal gene. And as a result, they do not have the disease, but they do have the potential to pass the disease on to their children. So let's label the copies of the gene that could be inherited from the parents as copy 1 and copy 2 from the father, and copy 3 and copy 4 from the mother. There's always 4 possible outcomes to the inheritance of this specific gene. Outcome 1 is copy 1 from the father and copy 3 from the mother. Outcome 2 is copy 1 from the father and copy 4 from the mother. Outcome 3 is copy 2 from the father and copy 3 from the mother. And outcome 4 is copy 2 from the father and copy 4 from the mother. So let's say copy 1 and copy 3 are the autosomal recessive disease genes, and copy 2 and copy 4 are normal genes. So outcome 1 has 2 copies of the gene that codes for the disease, and therefore this person would have the disease. So outcome 2 has 1 copy that codes for the disease and 1 normal copy, and therefore this person is a carrier, just like their parents. Now outcome 3 again has 1 copy that codes for the disease and 1 normal copy, and therefore they're a carrier as well. Now outcome 4 has 2 normal copies of the gene and therefore is free from the abnormal gene and doesn't have the disease and is not a carrier. Therefore, the children of these parents have a 1 in 4 chance, or 25% chance, of having the disease, a 2 in 4, or 50% chance of being a carrier, and a 1 in 4, or 25% chance of having normal genes. So by doing this, you can calculate quite easily the risk of the children either having the disease, being a carrier, or having normal genes. So now, finally, I want to give one scenario that illustrates calculating the risk of passing on an autosomal dominant condition. We can use the same method that we did for autosomal recessive conditions. So for example, let's say we want to know the possible outcomes where one parent has a condition and the other parent is disease-free. We need to know whether the affected parent has 1 or 2 copies of the gene for the disease, because either way they'll have the condition. So we can find this out through either genetic testing or asking about the affected parent's own parents. If the affected person had 1 affected parent, we know that they only have 1 copy of the disease gene, However, if they had two affected parents, it's possible that they also could have two copies of the disease gene. So for this example, let's assume they only have one copy of the disease gene. So again, we start with 4 copies of the gene, 2 from each parent, and let's assume that copy 1 is the gene for the disease. Outcome 1 is copy 1 and copy 3. Outcome 2 is copy 1 and copy 4. Outcome 3 is copy 2 and copy 3. And outcome 4 is copy 2 and copy 4. So therefore, outcome 1 has the disease, outcome 2 has the disease, outcome 3 has 2 normal genes, and outcome 4 has 2 normal genes. So therefore, the children of these parents have a 2 in 4 or 50% chance of having the disease, and a 2 in 4 or 50% chance of having normal genes. So I hope this explanation has made it a lot clearer about how you calculate the inheritance of these single gene diseases. If you like any more information or you're a bit confused, please post in the comments below. I'd be really grateful if you could like this video, subscribe to the channel for more videos.
Okay, so hopefully that kind of helps with some of these disorders a little bit, understanding kind of how a Punnett square calculates it, that sort of thing. So autosomal dominant, again, the autosomal part is talking about basically that these are not characterized by the sex genes, but the other genes with an issue. So inheritance of just one of the dominant alleles, again, allele, just a gene representing kind of the parent's information, can cause the disorder. So the patient only needs to carry that allele. So there's really no carriers here, because as soon as the dominant gene is a part of genetic makeup of the child, then they're going to have the disorder. If they are if they don't have the dominant gene, then there are no carriers, they're unaffected, they can't transmit it. And some of these conditions maybe have a delayed basically genotype or a delayed phenotype. It may be passed before there's diagnosis or treatment. And one of the common ones is Huntington's. Huntington's usually doesn't develop until adulthood. So it's hard to know that you have it until symptoms develop much later in adult life, or if you do some pre-screening and that sort of stuff, the only way you would know. So what type of disorder is characterized by the inheritance of one allele causing disorder? You only need one allele to cause the disorder. So think about kind of the Punnett square. Let you pause and think about that. Okay, the correct answer is this is the dominant disorders. For autosomal recessive, they would need both the lowercase letters. They need both those recessive genes to have the cause the disorder. Now, again, in autosomal recessive, they can be a carrier with one big and one little and have that recessive gene and pass it on. But in autosomal dominant disorders, they cannot, because the dominant gene takes over. So if they have that dominant gene, they're going to have the disorder. If they don't have it, then they're not a carrier. They don't pass it on. So the answer to the inheritance of just one allele causing a disorder is dominant, autosomal dominant. So X-linked dominant disorders. So heterozygous males and females are affected. Tends to be reduced in females. It's a fragile. There's a couple of fragile X syndrome is the most common mental retardation, mental handicap in North America. It's called Fragile X syndrome. It's a cognitive deficit, learning disorders, underdeveloped. Males and females can be affected. So basically, X-linked means that it's affecting the X chromosome, which males and females have. Males have XY, females have XX. So it can affect both males and females. It tends to be more prevalent in females because females have two X chromosomes, right? So kind of double the chances. to have Fragile X syndrome. And then there's X-linked recessive disorders. And again, Fragile X is an example of an X-linked dominant, so they just need the one allele from the parent, and bam, they're going to have it. Again, this is X-linked, so this allele that's recessive that causes disorder is carried on the X chromosome, not the Y. So sometimes females are going to be carriers of this. Sometimes inherit skips generations. Duchenne muscular dystrophy and hemophilia are examples of X-linked recessive disorders. So tends to be more in line with that. Basically, the matching male doesn't have the affected, they're not going to have the affected gene on the Y, but females, because they have two X's, have the potential to be a carrier or be affected by this. So chromosomal disorders talked about on page 576. Some examples of this are, and these are pretty neat to know, I would say. So Down syndrome is typically, you've heard before, its technical name is trisomy 21. So it may be because of basically translocation, just a movement of the or loss of the chromosome. Basically, an individual with Down syndrome has 47 chromosomes instead of 46. There's like an extra chromosome, basically, is what Down syndrome is. And then Turner syndrome is known as XO. Affects females predominantly. Turner syndrome basically is they're kind of missing the Y chromosome, but they're all, which makes them female, but they're missing another X. And so it affects female reproductive development, fertility, those kind of things. And Klinefelter syndrome is XXY. It's an extra X chromosome is present. This typically does cause infertility as well, kind of messes with sexual development, very similar to Turner syndrome. But Turner syndrome, they're just basically missing an extra X. Klinefelter syndrome, they're having an extra X. And then Down syndrome is having an extra chromosome. So, which of the following is not a characteristic of an X-linked recessive disorder? So, this is a tough one, but X-linked recessive. I'll let you pause and try to think about it. So, not a characteristic. Only males can inherit the disorder. It's definitely not. It's much more in line with females are more involved in X-linked recessive disorders and dominant disorders because of the X-link and the females being XX on their sex chromosomes. Multifactorial disorders. These are genetic influences mainly combined with our environmental factors, and they can be kind of, again, multifactorial is basically just there's multiple factors that contribute to it. So cleft palate, congenital heart disease, congenital hip dislocation, even type 2 diabetes is technically a multifactorial disorder because type 2 diabetes does have a hereditary component. You're more likely to suffer from type 2 diabetes if you had parents that suffer from type 2 diabetes, but also obviously diet, exercise, those kind of things play factors into diabetes development, especially type 2, not so much type 1. Therefore, it makes it a multifactorial disorder because there's multiple factors involved. And then overall, kind of rounding out developmental disorders, these would be more in line with going on those other factors for women, avoiding, things in their environment, drugs, chemicals, radiation. TORCH is an acronym for maternal infections that can result in congenital anomalies. So that's what we do prenatal screening for those kind of things. Torch is just a diagnostic tool. You don't have to necessarily know the acronym, but it's toxoplasmosis other, which would include hepatitis B, mumps, rubella, varicella, gonorrhea, syphilis, rubella, cytomegalovirus, and herpes is kind of what torches stands for. That's not a need to know. It's almost a nuts to know. It's kind of nice to know, but it's just an acronym to kind of help predict infections and those sort of things and screen that because they can lead to genetic disorders, that sort of stuff. And so again, drugs, chemicals, radiation, infections, those are all the teratogens, basically, that we want to avoid for females that are childbearing age or people that are pregnant for sure. And this just impairs what the book is calling the organ genesis. It impairs the development of the organs in the fetus and in utero. So diagnosing these, there's a lot of tools we've developed over years, but there may be testing even prior to conception to kind of narrow down the alleles and those sort of things. There are also you can test in the newborn, first trimester pregnancy. Family history is important for these things. Any previous, if someone's had a previous child born with any other abnormality, that would be a kind of a clue to check for the second pregnancy, what's going on. And then there's certain ethnic groups and regions in the world that have a high risk for specific diseases. And then women in older childbearing age is always, so over 35 years is always the risk starts to increase kind of after that age bracket. And again, there's blood tests, there's in utero testing, amniocentesis, chorionic villi assay. There's a lot of stuff, neonatal testing, those kind of things. So you don't necessarily have to know all the testing. What's a common environmental factor that can lead to a congenital disorder? So this is a pretty obvious answer, but the rest of these answers are not really environmental necessarily. So exposure to drugs, chemicals, radiation during pregnancy, that would be an environmental factor. So talking about some of these disorders specifically to round out this chapter, Down syndrome has become fairly common. Again, this is technically named trisomy 21. Basically, the 21st chromosome, they have an extra one. And so instead of 46 pairs of chromosomes, people with Down syndrome will have 47 pairs of they'll have a 40, excuse me. Instead of 46 chromosomes, not pairs, 23 pairs, they'll have a 47th chromosome affecting around the 21st one. It affects mental development, physical development. One of the risks they've highlighted is just increased maternal age. There are screening available, amniocentesis, those kind of things. Characteristics of someone with Down syndrome. There's some pictures in the book on page 581, but smaller head circumference, rounder face, flat facial profile, slanted eyes, epicanthic fold, which is kind of talking about the fold of the brow, large tongue, high arch palate, small hands, single palmar crease, short stature, loose joints, just overall delayed sexual stages, delayed developmental stage, and then obviously cognitive impairment ranges from mild to major. So there are plenty of people with Down syndrome that are fairly functional, and then some that need a lot of assistance as far as their cognitive ability. So it kind of ranges. And then just delayed physical development, sexual development. Males can be infertile. There's a higher risk for them to be infertile and lower rate of conception for females with Down syndrome. So, what's a risk factor that they have identified for Down syndrome? I mentioned this kind of quickly, but this is kind of the one thing they've definitely identified as a risk for developing Down syndrome. I'll give you a second to answer the question. Okay, answer is advanced maternal age, so greater than 35. It's potential for a higher risk of this. And these are some of the pictures from the book, kind of the head shape, the epicanthal fold, so the eyebrows, the brow, slanted eyes, just to give you an idea there. Okay, that is chapter 21, congenital and genetic disorders. It's a tough one, so obviously if you got any questions, let me know. Thanks for watching.